I actually hit share screen, right? I'm just like partially here today. High schools are closed, my husband's home, and he is not helpful to have around the house in the morning at all. Okay. So, last time we met, which seems like a little while ago now, we had just finished, being aggressive, discussing the basics for DNA replication. Okay, so what is it we need to make DNA replication work? All right, so our very first thing was something to copy. So we talked about needing that DNA itself, right, as a template, and we talked about DNA being semi-conservative. So we split that parent molecule in two, and we talked about being semi-conservative. We'll talk about this a lot, about how we can take that dark blue parent strand and, right, since everything, okay, does allow for complementary pairing. In other words, we can always do the opposite. So A will always pair with T, C will always pair with G. Okay, we can fit that together, right, like a puzzle, which allows us to build a new daughter strand. So when we say semi-conservative, of course, in that case, we mean we have one conserved piece, one OG, if you will, and one piece, the light blue piece, that's brand new. So that's the thing we're copying. And then the thing we talked about at the very end, okay, were our workers, and we're going to spend a little bit of time now going into more detail on these. Okay, but things that do the copying. So we talked about primase. Think primase like a primer, like your paint primer, right? So this goes down first. It does a little bit of the work. So we talked about them laying down like a runway, okay, an RNA runway. But polymerase, okay, does most of the work, right? This is like our construction workers. Even has DNA in its name. Okay, then we need, sorry everyone, I have like a frog in my throat this morning. I know that must sound absolutely annoying, particularly online, as I just like into the microphone. All right, then we need the stuff to actually do the building, right? So these are our materials. So you might think about this stuff being like the tar, right? The cones, the shovels, right? Everything we need to do a good construction job. Okay, so if primase is laying down our RNA runway, we need a little bit of RNA. Okay, and then remember we needed nucleotides. And we're talking about nucleotides here. These are the DNA nucleotides as we've defined them. Okay, this is all review, right? This is where we left off at the very end of class, last class. So what we want to go do now is start looking at some of the details of how do we actually do this. Okay. So we're going to break this down into ultimately 3 steps. We're going to start with initiation and elongation, okay, to start and to continue. And then eventually, if you remember our original roadmap, we'll have termination, right, to stop. All right, initiation. So how do we start the process of DNA replication? Okay, so the key here is we create what's called an origin of replication. Okay, so origin is one of those fancy words that means beginning. right? So we have, right, these two strands, and DNA is always going to be shown in blue when we do our drawings. So here are our two strands. Okay, so we're going to pop those two apart in a spot. Remember, DNA is very long, so when we open them up, right, we just end up with this little bubble, right? The whole thing can't get opened up at once. Right, we're just going to end up with a hot, tangled mess. So we're just going to open them up in one little spot. Okay, so you can see how that one little spot, when you pull the two strings or strands apart, right, it kind of looks like a little bubble. It's kind of a cute little name, replication bubble. Okay, so now in this bubble, we have some space in between our two strands. So if we look at the corners, right, or the edges of those two strands, we have what are called the replication forks, okay, which I've now indicated here in my red circles. Okay, so what is happening at our replication forks is we're going to continue to unwind. Right, we'll continue to pull apart our strands as we go. Right, remember our goal is to replicate the whole DNA sequence. So even though we're going to start here, we're going to eventually move along the whole thing. Okay, so we're going to pull that apart and move out in that direction. Same thing this way, right? We'll pull this apart and move out in that direction. Okay, these terms feel okay? Everybody see what I'm looking at when we're looking at this? Okay, so we're just popping it open in one spot. There's our bubble. Okay, and on the edges of our bubble is where we're going to continue to move out from, right, where our strands are still connected. Any questions so far? Okay, so let's look at the diagram that we have then, right? So here's our replication bubble. Or origin of replication, as it's labeled here. Okay, so then each of these corners would be a replication fork as we defined it on that last slide, right? Okay, so how do we actually make DNA be replicated? All right, first thing that needs to happen, remember primase was the first thing that needed to come through. Remember primase, primer, right, like paint primers. The first thing you have to lay down. All right, so primase, ASE is telling us this is an enzyme. We saw that a couple of slides ago. It's a good thing to remember. All right, we learned this when we did our biomolecules. So primase is going to come down first, and it's going to lay down an RNA primer. And an RNA is not DNA. So we've laid down something kind of funny. But this is going to serve, and in our drawing you can see it like the red, little red teeny strips here. I'm going to clean up a little bit of my drawing here just so we can focus on what I want to focus on now. Okay, so any of these little red teeny strips are our primer. that primase has laid down. Now this is going to serve as a runway for us. This is going to make it much easier for the next guy, DNA polymerase, okay, to land and do his job. All right, so step one, primase comes in, lays down that RNA primer runway. All right, now the key here for everything that we do ever, pretty much, is that everything we build starts with a 5' end. I'm not drawing, I'm erasing. There we go. And moves in what's called a 3' end. So everything has a direction. They're like Lego pieces, right? They have a bumpy top and like the open bottom. We all kind of picture what a Lego piece looks like. Okay, DNA strands work the same way. Okay, they have directionality to them. And we have the 5' end and the 3' end. Okay, so like Legos, we can continuously hook pieces together. My Lego pieces drawing definitely need work. We can all get on my bus, right? Okay. Now here's the key piece. Okay. Really important thing to remember, if you only remember one thing, right? I can only add to the 3' end. Okay, we can only build on a 3' end. So if we imagine this being Legos, we can only put them on the open bottom. Okay, so imagine having one of those like smooth topper thingies. Remember having those with my Lego sets. They was aesthetically pleasing, right? So we can't put anything on the 5' end. We can only build to the bottom. This is unidirectional construction, which is kind of nice. We don't like to learn this in one way. Okay, but that also means we need to be very careful. So when I say runs 5' to 3', that means I start by building. Right, I've created a 5' end, right? And then I'm going to build everything in a 3' direction, right? My empty bottom Lego direction. And as long as I have a 3' end, I can build in that direction forever. And remember our rules of opposites, right? Here's the 5' end on my template strand. Remember, everything works in complementary. So the 5' end on my template strand is going to match up opposite to the 3' end on my buildy strand, right? Just like A pairs with T, 5 pairs with 3. That kind of rhymed, eh? So this is another way, right, that complementary, all opposites business. So we're going to only add on the 3' end. Can we remember 5 pairs 3? And if we remember those 2 rules, okay, the rest of this, just like everything else we've done, is just putting a puzzle together. Okay, we can get a blank slate of anything, and it's just putting those 2 rules down. We can piece it together. Okay, so if we look at the bottom half of this, we see we have the same situation. It's grab a different color. So here's my 3' end. You see here I have my primer. Remember, my primer is my cap or my start. So that's my 5' end. All right, so here again, I've matched a 3 and a 5' start. But 5 and 3 have matched together. Okay, and if I look at the direction of that bottom one, I'm moving this way. And I know from my rules, only adding in the 3' direction. Okay, so this must be the 3' direction of my building strand, that light blue strand. And so here again, I see 3 matched with 5 on those 2 strands. So we've got opposites.
So someone asked in the chat, so is the template strand split in half and one strand of the copied DNA is made in the opposite direction of the other side?
Yes. Assuming I've translated, gotten all of that correct, right? So as we open this up, right, we see these are going to be made at the same time and in opposite directions. Right, because our original template strand, right, 5 here, 3 here, is opposite pieces. All right, so everything is opposites. So when it comes to anything DNA, that's pretty much our rule. We have that nice word complementary. Okay, but all it means is opposites or mirrors. Now, when we're actually building that 3' end, can we see that this is in baby blue? Okay, so very small portions, as we said before, are the RNA, right? The primase did that. That's our baby Runway. That 5' top is very small. Okay, so that's the primase setting up that 5' topper. That's all RNA, and we want that to be very small. Right? We're supposed to be making DNA, for heaven's sake. So all of this baby blue then is the DNA, and so most of that then is being done by DNA polymerase. Okay, putting down actual DNA nucleotides. Okay, always, right, always on that 3' end. All right, there was a lot here, right? So the key is focus on the rules, and they'll help you get through any problem. So, are there any questions on all of the rules we've established? Okay. Well, let's go ahead and why don't we take out, no, I'm lying. We're going to do this first. Okay, so the last thing I want to add then is, despite the fact we just keep using the word DNA polymerase, okay, this is a big job. So we still have RNA primase laying down that primer, but there's more than one polymerase. Sort of like when you drive by a construction site, there's not just one type of construction work. Okay. Now, I want to bear in mind that the way they're numbered here is not the order in which they do their job. Okay? It's honestly, they're numbered in the order they were discovered. Okay, so this is one of those weird things. And you'll encounter that a lot in science, right? We can only name things the way we find them. So we happen to have found DNA polymerase 1 first. It actually goes last. So if we think about the order of operations here, the first thing that's going to happen, there we go. How far did we get in? 25 minutes before I did it. Not bad, right? Let's try that again. The first thing that's actually going to happen, right, is not one of these guys. It'll be our RNA polymer, or primase. Everything's a polymerase now, right? That's going to be number 1. We're going to lay down that 5' end, lay down that primer. Remember, primer is first. So we don't want to forget about that, even though we're focused on our polymerases here. All right, after that, we're going to come through our big dog, hardest worker of the bunch, is DNA polymerase 3. This is why I've labeled it as the main enzyme. Okay, this is the construction worker that's doing by far the vast majority of the work. Okay, if we think back to our previous slide, this is the one that's putting down all of that baby blue. And remember, we're always adding on the 3' end. Okay, the open bottom Lego. Okay, but like any job, this cannot be where it ends. Okay, so the next step that we want to do is come through, pull out that primer. All right, because remember, this is DNA. So now that we've got everything added, okay, we can pull out those landing strips. And we want to change them from RNA to DNA. Because when we're done, okay, everything needs to be DNA. Right? Remember, A binds with T, C binds with G. So we're going to close our strands back together again. Okay? We can't have any U's in there, right? Or the strands won't ever close back up. They'll all be bunched and gappy like a bad old zipper. Okay, and then lastly, okay, just like any last thing you do, okay, we have the repair function. Okay, just like when you write a paper, the very last thing you do is check it. Okay, I check for errors. Okay, so we kind of teased at this sort of thing when we were talking about like meiosis and mitosis, right, with their checkpoints. So this is one of those checkpoints. We're going to go through very quickly, right? All of these things happened quite fast. They're very, very good at their job. But they also did them very, very fast, right? 500 bases a second kind of fast. Okay, so he's just going to run through, or she, and he's going to flip there and double check that everything matches like it's supposed to. And if not, right, we're going to pluck out the offending base and put in what belongs there. Remember, DNA is very important, right? It has a very important job. So we want to make sure we don't screw it up at the last second. All right, so it's like running spell check at the end of your paper. Or using Turnitin. All right, everybody's job makes sense? Right? So we still have a big dog. Okay, but there's more workers along here that are sharing some of the workload. Okay, now what I want you to do is take out that in-class activity we were working on before that has char graphs rules on it. And what we're going to do is start building a replication bubble. All right, so we're going to work on making sure we understand what all these terms mean. And what I want you to do, and we're not going to use all of our enzymes, right, is draw it kind of like we did a couple of slides ago, where we had our bubble. We have a 5' and a 3' end. Okay, I want you to think about the roles of primase, DNA polymerase 3, and DNA polymerase 1. Okay, so again, in this particular case, either using more than one color, right, to dictate the difference between RNA and DNA, or some kind of pattern difference is going to be really key. I did myself, I did not. Give myself a space to work on. One of these days. So remember, as you're setting this up, the key to success is a questionable replication bubble, okay, is make sure that you're identifying your 5' and 3' ends, right? Remember, opposites are the key. So I'm just going to identify 5' and 3' ends. So my 5' for this strand is going to be up here. The opposite of that makes this its 3'. 5 is going to be opposite of 3. 3 is going to be opposite of 5. Because these are my parent strands, right? So I've made them dark blue to try to match the colors that I've been using in my lecture so far. So I haven't built anything. This is just my initiation. This is just my replication bubble, my origin of replication with my forks. Okay, but I can identify all of the complementary parts on those, and that'll help me establish when primase goes in, okay, and sets up its 5' end. What direction then is it going to go in, right? Because where it can only add on a 3' end, so what direction is that 3' end going to go, right? Because 3' is going to match with the 5' of something. So let's try that. So I'm just going to do a little bit, but you need to finish the whole thing, right? So if I put in a primase, let's say put in a little RNA. I'm just going to keep all the same colors we've been using. So this is going to be my 5' end for my built strand, right? So here I have 5' opposite my 3'. Are we recording? Thank heavens. Yeah. Girl, I'm just not there today. Okay, so given that, let's go ahead and erase this. Is my strand going to build to the, you're right as well, right? Okay. To the right or the left? So what's going to be 3' for this guy? For the built strand. Let's pull up baby blue here. To the left, right? So why left? Oh, rats a frazzle. Now I erased it. Put some DNA in. And then DNA Paul 3, that's a 3. So this is my 3' end. All right, so we are going this way. All right, so remember my 3' needs to be opposite of the 5' for the template strand, right? Everything wants to be opposites. So here's the 5' for my template strand. My 3' wants to be by that. All right, if I'd gone to the right, let's pull up a different color here. If I'd gone this way, right, my 3' would have been with the other 3'. That would not be opposites. Does everybody see that? So we could have done it, but we would have immediately been able to stop and check ourselves and say, oh, shoot, that's not complementary, so we don't want that to happen. I need to go the other way. Okay, now everybody see sort of the benefit of this opposite system? Okay, so it kind of feels like a lot at first. Once you get that rule down, it makes a really nice check system. Because I don't even know any of these nucleotides that are in here. Okay, we'll talk about that when it comes to transcription and translation. Right now, I don't care. Okay, all I really need to know is I'm going in this 3 direction, and that's enough to know what I need to do. I know in the back of my head all of those bases are also opposites. Everything is opposites. But I don't have to deal with that.
So somebody asked, will primase be on the bottom strand as well?
Correct. Absolutely, right? So you would have two of these, right? One going across the top and one going across the bottom in the opposite direction. All right, and I would have DNA polymerase 1 removing the RNA on only one of them. Otherwise, your drawing is probably going to get really, really messy. Okay, this helpful, right? Ideally, all my in-class activities are helpful to you, but is it helpful to kind of see how this goes together? Right, having to make these decisions on the fly on what we're doing. Right, any questions about the pieces we put together here? Okay, this is it. This is going to finish this in-class activity. So what I want you to do, and I'll unlock it, right? But as always, right, make sure that you have finished this. This is not finished, right? As always, we don't finish them in class. We start them together. So finish all of these pieces, and then I'll unlock the Dropbox. You'll have till tomorrow, like always, to turn it in to the in-class activity Dropbox. Okay, which means if you are stuck or are frustrated, you have that time period to get help on it as well. Are those pups for me or?
So somebody asked a question. It said, will RNA be on the bottom strand, or is the bottom strand still DNA?
So the bottom strand will look identical to the top strand, right? So primase will go down on the bottom strand. It will also lay an RNA runway, right, creating that 5' end. And then DNA polymerase 3 will come in and create, right, 95%, which is that DNA replication. So yes, RNA will be on the bottom. And yes, it will be mostly DNA. So the bottom will look exactly like the top, right? Both strands need to be replicated completely and fully, and they'll use the exact same process. Keep in mind, guys, all we're doing here is S phase from interphase. Right, duplicating that DNA, this is just what we're doing, right, is S phase. So we're looking at how does S phase occur. All right, any other questions? All right. Okay, so in case my drawing is not great, another way that can help you look at these enzymes, putting things down. All right, so that's the core and how this is happening. I want to take a look at a few other enzymes that help make the work go. Okay, in other words, help stabilize. Okay, so if we consider the unwinding action, okay, if I were to take like a cord and yank on it and it starts to unspin or untwist really fast, a lot of times, I was circling, what you get is this twisting action. Right, so we can kind of imagine headphone cords. You know, AirPods are a thing now. Right, but when you try to untwist headphone cords or necklaces, right, when you try to untwist necklaces, those wristwatch chains, right, any of those types of things, right, when you pull on them, if you're not careful, right, sometimes they just twist up harder. And so this is the things we run the risk of when we're unwinding DNA. So we have a series of enzymes that work in cahoots together to make sure that as we're pulling our two strands apart, that they don't end up knotting up. Okay, so enzyme number 1, helicase, is just a zipper. And that's what I kind of like to think about this one. So that's just this guy here, right, sitting at the edge of our replication fork and works exactly like a zipper does. Okay, moving along and actually doing the pulling apart. Right, so when you pull the zipper on your jacket, okay, it makes 1 strand, 2 strands. Right, 1 zipper, 2 parts. And that's all helicase does. It's our zipper. Okay, and it doesn't care if it gets twisted or not, right? Its job is one job, unzip. So everyone else's job is to make sure that right, helicase unzips, nothing gets twisted up. So next up on our list is the mouthful, single strand binding proteins, SSB. Okay, very useful name. Very difficult to say. Okay, but the key is as you unzip this thing, kind of like if you think about like pulling tape up from the roll. Okay, just like tape. Okay, our DNA strand likes to, right, stick back down together, right, until you've actually filled it in. So once we've pulled this apart, okay, we put things in the middle here to keep it from sticking back down together, right? To keep that tape from flopping back down on itself until we replicate it. Okay, so that's what single strand binding proteins are, and they kind of tell you that. They sit on a single strand. Keep them single. Right? They de-stickify everything. Right, and lastly, just going to get all these mistakes in today, aren't I? Okay, topoisomerase, right, which is a whole group or class of enzymes. And these guys are going to deal with the twists. Okay, so we're going to picture this guy out here. So this is going to sit on the double-stranded portion out in front of our replication bubble. Okay, so this is like when you know you have a big twist in your earphones or your necklaces or your wallet chains. So before you pull on them, you have to kind of unravel and untwist them as you go. And that's what these guys are doing. They're making sure you don't end up with the quadruple knot. Okay. Or if you've ever dealt with like power cords, right? How for whatever reason, every time you pick them up, the cord wants to like wrap up and up and up on itself. So you always have to kind of figure out what way it's going and whip it a couple times in the other direction. Right? Everyone's kind of had to do that or like old school video game controllers. You got to whip them in the opposite direction because they get all bound up. Okay, that's what these guys are doing. Okay, they're basically just untwisting in the opposite direction so these things don't get quadruple looped up and turn into giant horrific knots. So what I have under here, DNA gyrase, is just one example of a type of enzyme within this enzyme class, the topoisomerase. Okay, so these are like specialists. These are all in addition to our primase and polymerases, right? Because these aren't laying down any nucleotides. Okay, they're just helping to make the work site safer. It's like OSHA. Okay, any questions about this? I'm going to clear this here. Okay, make sure, and I'll change the inbox here in about 5 minutes, that you turn in your in-class activity and that it makes sense to you, right? Like all in-class activities, these are meant to be helpful and they're really good study tools, right? On the last exam, meiosis was the worst question set. Okay, which makes me think that either those students did not finish the meiosis in class or didn't use it as a study tool. So make sure that you're really using these as a valuable tool. All right, as always, once you have finished your Socrative, you are good to go. Have a wonderful day.
I was just wondering, how many points do you take off of our attendance grade for each day that we miss?
So it's only for unexcused absences.
Okay.